To step 3, defining relevant indicators. There's a bit of content in here, so take it away, Chris.
Yeah, big focus on this is temporal and spatial variability and access issues. And I think the temporary waters guidance document, does quite a good job in this space, pretty good job. And the main guidelines themselves do a much better job as well in terms of some really good, input from CSIRO in terms of dealing with variability in a statistical manner. So, all right, so when we select our indicators across the PSCER framework, we're going to be faced with, as I said, the high spatial and temporal variability and the likelihood of access difficulty. So with this in mind, variability, of course, has the problem of, the potential to mask water quality inferences. So the places are known as on having a very good balance of indicators across the pressures, stresses and receptors, and that's going to increase confidence in our assessments. So we need direct and indirect measurement approaches to address these issues. So yes, so accounting for variability and access, we need good information on both the water quality and the non-water quality pressures to separate cause of impact. So we've got our typical water quality related ones, that could be, loads or, aerial extent of acid sulfate soils, for example, sediments. The amount of land disturbance quantified. For mine sites, whether or not water is treated and, leads to a conversation of whether or not, water treatment is better options in terms of managing wastewaters and, pesticide or fertilizer applications. They can be quantified as well at a landscape scale. Then we've got our other pressures, as we've said, all the livestock and feral animal damage. And again, those sorts of things can be quantified these days with new methodologies. So then our stresses and ecosystem receptors should be integrative. And for stresses, it's good to be able to be measured for as far as possible with equipment that can be left in situ to overcome that sort of access problem. So in that case, we've got, continuous monitoring or auto sampling. Sediments are very well placed to be integrating the effects of past exposures and can be collected in a dry phase as well. Direct toxicity assessment, that was integrate the effects of whole mixtures, whole waters, and all the mix of contaminants associated with the wastewater. And, bioaccumulation, macroinvertebrate assemblages. In the guidance document, we mentioned propagule bank and refugia and the hypereus of the dry bed as all useful indicators of receptors and that have that integrative capacity. Okay, so this is where I was going with mentioning some of the more statistical aspects of things. So from my perspective, the guidelines perspective, good characterization of stresses and ecosystem receptors over the whole, all phases of the hydrograph, and different rainfall years, I think is important in study design. The more that we can throw into models, I think the better explanatory power you can get out of it. So multiple sites should be incorporated as far as possible. And maximizing as well just the actual or potential disturbance gradients. That's invaluable in terms of looking at some sort of biological effects approach to looking at guidelines. So nowadays we've got very powerful statistical models that we can start to make just a lot more sense of variability in the data. And these methods can be used to develop typologies, classifications, and from those typologies and classifications, we can start to partition the data into components that we can then treat separately, or they might be continuous in some sort of regression relationship, so that we're looking at a continuum and some sort of departure along that sort of model. So as I said, these models and classifications can lead directly to guideline derivations, and you might then have cloud points that you've measured after you've derived these models, and outside of these relationships, you can start to infer impact. Okay, now in the guidance documents, there's a very comprehensive appendix, and it's really worth looking into. It'd be, with some of the new stuff that Andrew, in particular, will be presenting later, we can, look to this appendix and update it a little bit. But it's a really good basis for looking at the pros and cons of different indicators across both stresses and ecosystem receptors. It, as I say, looks at the strengths and pitfalls and the applicability. As I said, there's a lot more advances made since we put this guidance document together, and that's, as I said, Andrew in particular will present on some of that. Alicia's going to contribute as well. So in the guidelines themselves, we've sort of got a checklist there to just, look through all these issues and see whether or not you've covered your bases, the type of pressure and associated stresses, your management goals, the level of protection, which we haven't really discussed much yet. If it's an area of high conservation value, then the guidelines by default suggest you will need more indicators, and particularly some early warning systems. The spatial scale of the studies will be important. Obviously, we're dealing here with inland waters, but there are saline systems as well. The ecosystem type, whether we're in the waters or sediments, what part of Australia we're dealing with. And lastly, there I've got emboldened there the diagnostic value of the mix of lines of evidence for each of the pressures that we're studying. And that comes down to the first part of weight of evidence, and that's looking at the quality of the evidence that we're gathering from the indicators at the outset. We don't want to be evaluating a monitoring program where traditionally weight of evidence comes in, if in fact we've missed key indicators at the outset. So I guess a key part of Australia's take on weight of evidence is having a two-part step. So at step 3, we've got for our 7 typical uses, a quality of evidence table that looks at all of the pressures, stresses, and ecosystem receptor types, and looks at, well, if you only measured this, what's the risk that you've missed something? Or, so on balance, what's the best mix of indicators across PSER that will give you probably a good outcome? And this is a generic tabulation, but as I said, it's for each of the 7 typical uses. We've spelt this out in a lot more detail. But basically, we've got on our left-hand side the merits of all of the particular indicators, And yeah, they're so obvious, but measuring water chemistry, it's a direct measure of the potential stressor. But on the other hand, maybe it's not that particular chemical that you've measured, and you may have missed something. And that's where of course, the biological monitoring comes in, and its ability to say whether or not, in fact, you have an impact at all. It's looking at the direct measures, the management goals themselves. But there's pitfalls there as well. In that, okay, we've found some sort of disturbance, but was that associated with water availability or feral animal damage or whatever? So at the end of the day, in these systems, the advice is to certainly be choosing indicators between stresses and ecosystem receptors in some sort of balance that best matches the situation.
Okay, so Chris asked me to present something about some of the work we've done at the Ranger uranium mine, because we have gone into different areas and we've got a big program of genomics at the moment. That's not changing. Go. 1, 2, 3, go. There we go. So uranium mining in the Alligator Rivers region is my job, and that's what the supervising scientist is there for. Some of you might have seen this couple of slides in my presentation for the conference, but I just wanted to show that we have been, looking at the rehabilitation of this mine, and many years ago, uranium was identified as a contaminant of potential concern for sediments in the benthic communities in those backflow billabongs that you can see sort of down and around that, which is pit 1 there. So we identified the need for a sediment quality guideline value to inform the closure of the mine site, and also as a target for operations and things like that. And we started this a long time ago, back in 2009, with a couple of pilot studies and the main experiment in 2012, 2013. But we started with traditional methods. We're going to count bugs in these sediments, and we just couldn't. It was impossible. So we went to a environmental DNA approach because it was pretty much our only option. So we did a mesocosm design for this. We took sediments from the field. We took them back to the lab. That's a younger me, spiking them with uranium sulfate. We did a lot of treatments to try and get a concentration response out of this. So less replicates, more treatments to try and model those, no effects concentrations. This was the boss at the time getting his hands dirty. We put them in these little baskets to look at the recolonization of the benthic communities into these sediments, deployed them for the wet season, pulled them out, and we sampled them for chemistry. There was a lot of chemistry done, but environmental DNA as well. And from that, we looked at the eukaryotes, so the micro zoobenthos that was in there, the metazoans, and also we looked at the prokaryotes, the microbial processes and the microbe communities that were in there. And to look at the function, we did a little bit of what they call shotgun sequencing. We did a bunch of hypothesis testing, threshold change analysis and things like that. And from that, we're actually able to come up with these guideline values. So those guideline values are actually rehabilitation standards as targets for the closure of that mine site. And like I said, it was the only option because we couldn't really count anything in those sediments. It was way too hard. I mean, we could have, but the resources that were going to go into it were just not feasible. We did include prokaryotes because you can do that with your eDNA techniques. And, biogeochemistry is often overlooked when you're deriving guideline values in environmental regulation. But in this case, it was less sensitive than the eukaryote responses. So I had lots of lessons. One of the main ones was it's really helpful to have no other options to get buy-in for these novel techniques, because it's like it's this or, you know, we go to sort of background levels or something like that, some other referential approach. There were early adopter headaches with it. We spent a lot of time on sediment chemistry, and that was really well done. But, you know, the eDNA techniques were still developing. Back in this day, we used a technique called 454 pyrosequencing. When we came to publish, we wanted to look at it in different ways. So it's still a bit of a headache, to be honest, but it was a great challenge, and it was really useful information. Linking structure to function is one of my bugbears at the moment. Like, you know, we see community changes, but what does that mean in a functional sense? We really did a deep dive with the bacteria on that, with the metagenomes and things like that. That's Brodie Sutcliffe. She was an absolute superstar and really dug into what the function looked like. And that was great, but it was still a challenge. And, you know, so if taxa change, what does it matter, especially when you're talking about microbial processes and even those micro eukaryotes, like what do they really do in a functional sense? And you get a little bit of an interesting data set from environmental DNA, but we're working through that with better bioinformatics, better data analysis. I saw some great stuff at this conference with people doing prokaryote sensitivity distribution and coming up with guideline values in some novel ways. And I'll be talking to them about their approaches and how they do it. But what this really also does, it really unlocks those components of the ecosystem that are particularly hard to characterize, but are probably really important when it comes to temporary waters, such as the groundwater. So again, like going to groundwater, the fauna can be very difficult to find and measure. So we did this underneath the sands of Magilla Creek. There's a subsurface flow during the dry season, and we've found a plume of contamination from the mine site that's flowing during the dry season when there isn't water in that system. So we used environmental DNA to detect those community changes and what they're responding to within the hypereos and the shallow groundwater of the system. And again, we used these data sets to come up with threshold analysis. This is a tool called Titan, taxa indicator threshold analysis, something like that. But a lot of people are using it these days because it actually takes change points for individual species, which has some, or OTUs in this case, operational taxonomic units. But that also gives you some power, but it also does an overall community analysis. So you can use these tools to actually come up with guideline values. We're understanding those sorts of tools more and more. Like I said, some really good stuff happening recently. So that will really start developing, you know, potential new indicators for these systems that have been hard to measure in the past. We've been investing in environmental DNA because it's getting cost effective, it's getting doable, and I'll talk more about it in the actual monitoring section. So I'll leave it for the moment. I'll invite Alicia up to the stage to do her potential new indicators.
All right, I got to remember what I put here. So just a quick sort of backtrack to how we got to this point. So, you know, 20 odd years ago, we'd do our sampling. A lot of sites would be dry, so those bits weren't represented in our river health monitoring. And here's some examples of places that we were targeting: the Gulf and Southeast Queensland. And at that stage, we were sampling with a stratified random design, some massive areas. So these are like 100 by 100 kilometre kind of cells, had no sampleable water in them. So when we were reporting on the health of the catchment, we're really just reporting on the health of the wetted bits and not the dry bits. And in a lot of the systems, the dry bits rule. So even where we did sample and some of those other cells, that might have been the only sampleable water in that whole area. So my PhD was looking at what lives in a dry riverbed, and particularly it was focused on terrestrial invertebrates and whether we could use those as indicators of dry riverbed health. So it's not kind of rocket science. The idea is if we went to a site and it was wet, you could use your wet indicator. If it's dry, look at some dry ones. And apart from terrestrial invertebrates, something like riparian vegetation condition could be sampled at any time. So that's something to consider too. And that should link back to your conceptual model. So if it's important, then, look at riparian vegetation, look at the stresses that could be impacting those ecosystems. So invertebrates are my bag. It's not a coincidence that I'm wearing a dress with tardigrades on it. Water bears, they're tough. They love a bit of desiccation. They can survive. And in case you weren't aware, dry riverbeds can have a huge diversity of critters. So there's a whole bunch there. Is my dot working? Everything from A to Z. And you get a whole bunch of spiders, ants, beetles in particular, and some things that only live in the riverbed. It is a unique system that's different from the riparian zone. So they might share some species, but there are some taxa that only live in a riverbed, and you think, what happens when it re-wets? Well, it's similar to where the aquatic things go. So there are some terrestrial invertebrates that can withstand inundation, and similarly to what happens when it's to the aquatic invertebrates that can disperse during dry times. During wet times, terrestrial things can fly away. A lot of these things are insects, but also spiders. And I was interested in salt lakes earlier because and I know in WA there's some special salt lake spiders, and I was thinking about those with what happens with those lakes and the spiders if that ecosystem's compromised. So we've started to use terrestrial invertebrates as indicators of ecosystem health, similar to what we do with aquatic invertebrates. We look at species richness, to some extent abundance, but also looking at particular groups. So it seems like ants and beetles and spiders are quite important components of dry riverbed ecosystems. And when we talk about indicators, there's a whole sort of checklist of things you should be looking at when it comes to them. So indicators should be something that's ubiquitous, that's species rich in general, and also has some sampling techniques that are so nice if they're cheap, but easy enough to do. And then when you take the samples back into the lab, something that's not too difficult to do. So with aquatic invertebrates, often the level of identification is at the family level, and we can do that with terrestrial invertebrates pretty easily as well. And ants, often to genus is pretty easy, and ants have been used in other environments as indicators of ecosystem health, particularly in, say, mining rehabilitation areas. What else did I want to say? When we look at beetles in an aquatic system, might have 30 aquatic families that you could potentially get. In a terrestrial ecosystem, that just explodes. Like if you're trying to identify beetles, it could be 300 potential families, but there's a suite of particular beetles that I've found that seem to fancy dry riverbeds, including carabids, your ground beetles. I think maybe that's all I mentioned. Oh, there we go. I'm just circling back to our pressure stressor response model there. Just like to point out this, we've got a generic list of stresses that we apply to the whole of Queensland. And then when we look at particular areas, before we do any of our assessment, we run a risk assessment workshop where we incorporate experts in the field to provide information on what they think the stresses are and how important they are and how much confidence they have. So a bit tiny there, but we've got things like introduced riparian fauna and flora, so weeds, things like organic matter, acid soil runoff, salinity, those sorts of things. So those are a generic list of stresses, but then when we come to do a particular area, like I said, we tease out which ones came up as the highest risk. So, you're looking down here. This is an example of a report we put together. So riverine condition assessment of Warrego, Karoo, Bulla and Nevine. And for each catchment, and we looked at, so in this case, we're introduced aquatic fauna, particularly fish, introduced riparian fauna. So that's your feral pigs and your cattle. And deposited sediments, one that came up has been important because deposited sediment can reduce the depth of water holes and the water hole persistence. So for each of those, like I said, we looked at the risk, we identified whether it's high or medium or low, what the current threat is and the condition. And for my interest, introduced riparian fauna was what I was interested in, and the condition was assessed by using those terrestrial invertebrates. So we could determine whether it's slightly disturbed or moderately disturbed based on that number of terrestrial invertebrate taxa. And deposit sediment and fauna, we use different indicators. So things like percent, exotic fish species and those sorts of things, but my bag's the terrestrial invertebrates. I think that might be all. Yeah, thanks. So, have you got any questions? Thanks.
Oh, just put that previous slide back, please, Alicia. I think a big knowledge gap there. So, yeah.
Ah, that one? No.
No,
that one
there. A big knowledge gap there down on the right-hand side is deposited contaminants. So, if you've got an acid pool and it dries out, what's the response of your invertebrates?
Yeah, totally. No idea. So that deposited sediment was really just.
I know that.
But
that's for me a real knowledge gap that we should pick up.
Yeah, no, that's great. Thank you. I think so we should be looking at, say, a lot of this, these unknowns should be linked to some targeted research. So whether that's through universities or whatever, I mean, looking directly at that link between, experiments, toxicants, invertebrates, how they're, whatever, how they interact. Yeah, totally.
Hey, Alicia.
Hey.
I don't know, I tend to look at environmental harm from spills, and we're finding it terribly hard because people don't go to investigate till 6 months after the spill, and then these are rapid systems. Is this something we could apply on a smaller scale, or is this mostly about catchment-wide assessments?
We use it for catchment-wide assessment, but I don't see why it couldn't be used in a similar way to aquatic invertebrates, signal or whatever else, whether there's something that could be developed for the terrestrial side of things.
So how long after it dries up do you get a stable ecosystem? Sorry, I'm a chemist.
That's a very good question. This is really early days stuff. Like, not many people have really looked at terrestrial invertebrates in riverbeds at all. And there is that sort of succession. So, once it's dry, you've still got some semi-aquatic things carrying on. And whether the community changes until you get an eventual kind of dry riverbed ecosystem that's different to, the moment where surface water completely disappears. But I don't know if it's, a month later it's unique, or 2 months later it's unique. That's definitely an area of research. Good question though. Yeah, it's really cool. Cool.
Thank you. Sorry, Chris. Because I'm taking notes, I just need to ask what that key knowledge gap that you identified was again. Okay, cool. Yeah. Yeah.
And
thanks.
Something that hasn't really been discussed is with the natural riverbed, whether it's, if it's cracking clay, that's completely different from cobbles or sand or whatever. So how those toxicants and things interact with those sediments must be completely different. We know cracking clay does kooky things when it wets and dries. Yeah, that's of interest too.
I have a couple of other questions, if I may. May I, Andrew? Okay, thank you. Andrew, with regards to the eDNA, and you state with eDNA techniques we're getting there, And I remember us saying the exact same thing 10 years ago for eDNA techniques. So I'm kind of wondering where we are with eDNA, community DNA techniques in terms of implementing with the routine monitoring.
Routine's a key word. So I think that everyone's looking at their specific contexts. We certainly are for monitoring, and there's a monitoring section on that. But people are out there doing it and getting really useful information with it for their own context and things like that. The appetite for a regulator's acceptance of that will vary based on their context, and it certainly helps that Ranger's in the middle of a World Heritage Area, and we need these guideline values for a very important closure, which I think is why it was taken up. So there's things that are being done now that are great and useful, but everyone will continue to optimize that as we go along. The technology is only getting better. There's certain caveats that everyone should know about the data sets when they are provided, such as for bacteria, like a community change doesn't necessarily mean that the whole system's functionally different, and that's a fairly big caveat. So I don't see regulators using that necessarily now, but as another line of evidence, I think it's important. But then you can go to your field effects and test things if you see something that perhaps suggests that there is perturbation.
Thanks. Okay, so is the fact that it's a rapidly developing technology, a bit like sort of drones, remote sensing, will that hinder its uptake?
In a regulatory sense, I think it's going to take some time to convince regulators, but with data sparse and your baseline biodiversity of a temporary water where if you can't get out there and if there's no fish in the system and it's based on your micro not microbial, but your smaller invertebrates and things like that, then you're going to have to use something a bit novel. I guess that's why we brought it up here, was because going out and trying to catch fish isn't necessarily something you can do. And even your macroinvertebrates, it can take time and resources to go and do that. We're investing in understanding whether we can take water samples and increase that efficiency so that you can do greater temporal and spatial sampling. And I think once you get that sort of power in those data sets and those temporal, eDNA works really well when you start comparing it to itself over time. And I think that that's going to be the power. So that's going to take time to actually develop those data sets, I guess, is the point.
I just want to say, in my experience, people care about things as they get bigger, and it's going to be really hard to get the ex-cops that run our regulatory side to care about bacteria.
Don't we know it, Chris? Anyway, yeah, look, but that's about the conceptual model too, right? So we've been having an interesting discussion in our context at the Ranger Mine about where that fits in. And a lot of conceptual models just leave it out. And it's such an important, keystone, for want of a better word, part of the ecosystem. It's like just leaving it out doesn't make a lot of sense. So yes, there's conversations. I totally agree. We get some very blunt comments about people not caring about bacteria, but people should care about biogeochemical processes and what they mean for the whole environment.
And also just biodiversity in general. So, you might not even say bacteria, you might just say diversity of life. I just had a couple of comments. So eDNA is awesome in a world where taxonomists are dwindling and we're losing that expertise. So you can, throw your sample in and out pops stuff and you didn't have to, well, necessarily identify every single thing to species. But also, I was going to mention something secondary. That's right. Excuse my ignorance, but how does like uranium affect DNA? Or does it? I don't know. No? Okay, cool.
It's a common question, radiation effects. We don't really even touch on that in the ecotox program that we've run for many, many years. A lot of people are shown it's a chemical toxicity. I mean, the level of radionuclides is so low that it doesn't really impact. So yes, there's that. Do you have a question, Tony? Yep. We've got plenty of time. Yep.
Yeah, I think about eDNA. Went to a recent AMSA conference and they were talking about eDNA in the marine areas. And, you know, the smallest sort of changes in the marine habitat, you get these vast changes in bacteria. You don't really know exactly what's causing it. And I guess that's probably the main issue with eDNA is you get changes, but you're not quite sure what that actually means.
Yep, totally agree. And that's an area for research, right? If we're going to go to the microbial processes, I think it's got to be about function. It's something that we're doing in soils at the moment. We're trying to link structure and function, and that comes with a multi-omics approach that we haven't. I don't even have slides on it, so you can take it all the way through to the functional endpoints using metabolomics. So looking at what metabolites are being produced, proteomics, what proteins are it. So, you know, what's there in a DNA sense isn't necessarily what's going to be there in a functional sense, but there's other techniques that are coming on now that are even newer than eDNA. Like eDNA has been around, you know, a decade or more. There's some really good examples of people using it for decision making, not necessarily in regulatory context, but in, you know, healthy waterway scores and things like that. And there's some good examples. And the more we do it, the more we say, the more we're finding that it's a really useful tool. So the more we demonstrate it in other contexts, I'm sure at some stage, I mean, we're investing in it because the promise, you know, is getting mature and getting more real. And what we hope to do at Ranger is actually demonstrate how to put it into a regulatory context. And then hopefully we can feed that into the water quality guidelines process as a demonstration of one way of doing it.
Thank you, Alicia. Just got a couple of questions for you with the dry riverbed invertebrates. It might be too early for this, or I might be even missing the point. Can you pick up dry bed impacts associated with wet phase pressures and stresses?
That's the thing I'm super duper interested in. So the condition of the wet phase, how that affects what turns up in the dry phase. So yeah, if the water quality was rubbish or whatever, does that mean that dries down, and then how that affects the terrestrial thing? So I think it does, but I got no idea how. Yeah, totally. But it's, yeah, excellent question, and I think it's definitely worth more research into. Yeah, thanks.
So actually a little bit related to that, are you looking for or able to find any links or relationships between wet phase and dry phase communities? Like, it's a certain wet phase community, will you see a certain dry phase community? Noting, as Chris said, that they're totally different communities, but can you see relationships?
So that's, we've got the data to look into that. So you're talking about when you say communities, like at one site, you could look at the aquatic invertebrates, you know, early in the year, and if they demonstrate that the stream's in good condition, then once it's dry, is it still in kind of good condition with the dry, like the terrestrial invertebrates? Is that kind of what you're thinking? Sorry.
That's all right. Excuse me, I'm struggling. I've got a frog in my throat and I've got to talk next, so I'm getting a bit worried. No, it's more about certain types of wet phase communities. Can you associate them with certain types of dry phase communities?
Sorry, for the benefit of online, I don't know. Yeah, sorry, but yeah, good question. We got to.
I can respond perhaps to Rick from a South Australian point of view. The wet phase water quality stresses, say the where you've got eutrophic, highly turbid waters. Our approach of assessing dry watercourses would clearly show that sediments that are very highly silted, highly organic, prone to sulfidic conditions in sediments, and being eutrophic conditions mean you, like I said before, pretty dense algal coverage. We're pretty confident in describing the dry riverbed and inferring that the antecedent wet conditions were highly eutrophic and highly turbid or full of fine sediments. So yeah, I can refer you to Peter Goonan to give you details.
Clive? We do have a question from Melissa online, another good one from Melissa. Thanks for sending them through. It might be for me, it might be for Chris, but do you think mapping species richness and abundance over time would be sufficient to provide an indicator of health over time, or could it be used to establish baseline conditions for an ecosystem? Are the species able to adapt to a climatic change, or do you find the rate of change to climatic events to be too great for some species to adapt to? I.e., would this be a suitable technique to establish trigger limits relating to maintaining species abundance and richness? Do you want to answer that, Chris? Well, I mean, I guess that's just one of the complexities that's difficult to, you know, it's probably going to be a theme that comes up throughout the day, you know, and one of those things people are going to be looking for advice on going forward. Yeah, that's a pretty good idea. Shelley's got a response though.
I just want to throw something out here at this point, and this is some work that was done at JCU by a PhD student back 2010 to 2012, working with Richard Pearson, and it was working in the streams feeding into the Burdekin River and looking at the macroinvert community, but year on year, the composition changed. So as a short answer to that question, that might be really challenging around that. Essentially, the functional groups didn't change. There was still that inherent functionality in the system, but the actual composition in terms of species tended to change year on year. If anyone wants a copy of those papers, I do have them back, and they can just send me an email and I can flick them through. I know she's working in WA now, so I can also probably dig out her email, but that was a work that was done back in the 2010s.
It's a process, it's a Moran effect, but it's a temporal concordance, but with macroinvertebrates and in our billabongs up our way, you get shifts over time, but they're quite natural and landscape level. Even the invertebrates in the contaminated water bodies shift at the same way across time as the reference water bodies. It's quite intriguing, but the separation is still there, you know, so that it's landscape set. So I wouldn't say it's, I would say it's a rather natural phenomenon.
If you've got a good reference site.
What's that?
With your reference sites, you know, I think, you know, some people in, sorry, I should be, in some contexts, it's really difficult to find reference sites like we have in the wet tropics. So you get into those really temporal places and trying to track that might be even more difficult, right?
And I was just going to add to it that Angela's just left the room, but the Australian Rivers Institute at Griffith and Stuart Bunn's team spent quite a lot of time in sort of early 2000s working on the founder effect. That if you look genetically at say all the diticid beetles in a pool, they'll often come from two individuals that got there first and enjoyed the pool. And then that'll have an effect on what colonizes that inundation cycle. And the next time it's another couple to get in there. And so these things, that's why in the guidance document, we spend a lot of time talking about that, that the stochasticity of these systems means you need to better characterize your reference conditions. You need more diversity of reference conditions to be able to encapsulate that variability within the natural condition within the system that you're concerned with. But it's an important founding principle.
Overlying that climate change, I think is just going to add a whole extra level of complexity in trying to, tease out effect from noise.
Yes, more data. We're scientists, we want more.
I just confuse us all, really. We still have another 5 or 10 minutes before, 10 minutes, call it, before lunch. So keep the questions and discussion flowing, or we can just break a little early. Clive?
This is a comment or perhaps question, we'll see how we go, about the conceptual model in the guidelines framework. And I'm wondering about, because it flows on from reference condition discussion, and given that reference sites are rare, I'm wondering whether or not we can, so the pressure state ecological response thinking, can that be mapped against what we would call a human disturbance gradient? So not have it black and white as in equivalent to or worse than reference condition, but reference condition, however you assess that, is, call it natural or, good. And then have biological responses related to or mapped against increasing human disturbance, because that's what South Australia is trying to do. So we've got a whole range of biological response measures. And so this might go a little bit to Queensland's Alicia dry riverbed sort of thinking in terms of what bug community you've got in dry riverbed. And the sediment or riverbed characteristics, are you able to describe the particle size distribution, the gravelness or the siltiness or the clayiness that's attributed to natural factors, or to what extent it's impacted by anthropogenic or human disturbance factors? Because, some riverbeds are going to be naturally silty. And most are not, but so we're trying to map biological response driven by the stresses according to that human disturbance gradient. And is that being applied elsewhere is what I'm wondering. That makes sense?
Do you have a mic?
Yep. Oh, I don't. Oh, that's questions for me. It's such a hard one because different communities will be living in the cracking clays or whatever compared to sand, compared to cobbles, and I don't know. I don't know how to answer that. Exactly. So sometimes if it's a sand slug or something and it shouldn't be sandy, but you're getting critters that are living in it, versus if it was cobbly but it's been silted over, compared to if it's naturally silty. No idea. I think all of this is just creating more and more questions than answers.
So Alicia, I'm just reminded of your very good paper on this very topic, that you had a number of disturbance variables and the impact of ferals and cattle was, a key covariate of the disturbance gradient.
So I guess I should just mention, for the benefit of people that don't know, we looked at a gradient of disturbance and based on So it was based on land use, like tree coverage from satellite imagery, but then quantified by actual measurements in the riparian zone and linked to feral animals. And we saw a very obvious decline in species richness and abundance of terrestrial invertebrates based on that those impacts of feral animals. Yeah.
Was it compaction or what was the actual mechanism?
Yeah, so if you saw those pictures earlier, it's that compaction of the sediments versus, I mean, we don't know. We need research to say whether it was, you know, the nutrients from them as well versus that erosion impacts of just cattle in general, then filling interstitial spaces, which are habitat for those invertebrates. So it's one of those things we found a response, but then linking it back to why it's actually happening, that's a tricky one. I should say early on in my PhD, so Stuart Bunn was one of my supervisors, and he suggested I sort of pretend I'm a cow and sort of stomp around and do a bit of experiments, compacting sediments, out, which I didn't do in the end. But yeah, it's targeted research like that, linking the actual impact to what it's doing is important.
Any other questions? We can move on. We are a bit ahead of time, which I think is a good thing. I mean, I could use a nap after a long conference. So if we finish early, I might do that. But let's move on to the next section just before lunch. We've got 20 odd minutes, so we just might call it where we get to. So it's section physicochemical stresses, steps 4 and 5, deriving this water and sediment quality guidelines. And